Okay, so I start with a few announcements. First of all, okay, try to keep up with the homework, especially when it comes to the reading. We are covering a lot of important stuff that a lot of times is covered in biology honors, but not in the depth that you need to cover it. If you're not reading, you should be reading. For example, you should have read metabolism. We kind of talked about last time we had a lecture about endothermic versus exothermic reactions. I didn't quite get to the whole role of enzymes yet, or to cell respiration, or to photosynthesis, but I did give you examples of biological reactions, which are endothermic and exothermic on that lecture. I also talked about the need for coupling of the reactions. But we'll go back to that in a second as I revisit that 2.A.1. But make sure you're keeping up with the homework. You should have read cell respiration. You should have read photosynthesis. You should have read mitosis and meiosis by now, actually. So make sure you're studying a lot of things. I forgot which ones do this week, but make sure you check on it. Also, let me talk about Lab 3 a little bit. Okay, so those of you who actually turned in the Lab 3, I actually finished grading it already. I haven't not graded a bunch of other ones that have been turned in, but Lab 3 was the last one I finished. So let me actually talk about what should have happened. Okay, so first of all, if you did Lab 3, you should have noticed that the genes, one was for collagen, right? One was for the sodium-potassium pump. One was mitochondrial DNA, right? And then the other one, what was the other one? Ubiquitin, yeah. Which is what? It's like, what does it do?
Oh, it destroys defective proteins.
Yeah, it works on proteins.
It has something to do with proteases, which are enzymes which digest broken proteins in the body. Now, if you look at ubiquitin or collagen, so collagen is a highly conserved core structure among higher eukaryotes, anything that has like cartilage. So as early as a fish, it's going to have collagen, right? Now, there's going to be some variety among vertebrates in the actual structure of collagen, but all vertebrates, by definition, you're going to have collagen. So that's not a good candidate to differentiate between an alligator and a bird. With me, I'm trying to say? Because they will have that in their common ancestor. So it's not something, this is a very key feature of that lab you're supposed to kind of grab at, right? It's not something that's going to help you distinguish between them, all right? Because it's something they share. Does that make sense? Like, it's like getting a sequence of DNA that's exactly identical and using that to differentiate between two things. It doesn't make sense, right? It is the part of DNA that's the most different that you want to look at, right? To make sure that now when you do one of them, I think you get like a bird. The other one you get more like an alligator. Ubiquitin is the same thing. It's a highly conserved core structure among all eukaryotes. All right? Sodium potassium pump, highly conserved among all eukaryotes. So all those genes give you information, yes, because one of them goes to Gallus, the other one goes to another type of bird, the other one goes to, but the mitochondrial RNA, which is a fastly mutating DNA, because it's in the part of it's basically inside of a symbiotic organism that lives inside of us that doesn't have the kind of DNA control mechanisms that we do to catch mistakes. So it mutates faster, right? So it gathers differences faster. Does that make sense? So, but that means if you have a lot of similarity on that gene, it means that you can't possibly have split too long ago. Does that make sense? Because if you are 99% the same on something that mutates fast, it probably means that it hasn't been so long ago that you diverge from that. Right? But if you're 99% of something that mutates very slowly, because it's a highly conserved core structure among all eukaryotes, that doesn't really mean anything. I have some interesting theories were proposed for the bird one, for the sodium potassium pump, which is most similar to the fruit fly. Right? So let's first talk about why they put that there, before I give you the final answer. Why they put that there? They put that there to make you practice that idea of paralogous genes. Of genes that, not paralogous genes, sorry. I mean, Oh, okay. When two things are in the same branch, we call it monophyletic, right? When something is on a side branch, right? So, let's see. Get a mark. All right. So, if you have a cladogram, right, and you have the derived characters which are causing the splits on the tree of life, so everybody in these branches is going to be monophyletic with respect to that trait, right? Everybody in those branches is monophyletic with respect to that trait. But that right there is paraphyletic to them because it doesn't have that trait. You know what I'm trying to say? So paraphyletic means the out group, while monophyletic means like the in group, right? So it's the other term that means the same thing. Polyphyletic means that there's something in this trait that's also showing up over there, and then you're not sure if these maybe should be monophyletic. Like heat regulation and a bunch of enzymes associated with regulating body temperature and metabolism in birds and in mammals. High degree of congruency, evolutionary congruency, which makes you question whether your tree is right. Which is why we talked about back when we were doing evolution that it's important to think about in terms of parsimony. All right, if you suddenly were decided to switch the entire tree to put the humans and the mammals and the birds together just because of that trait, then you would have to explain another 30 traits which are they're different about. Everything I'm saying? And that would make less sense. So you pick the simplest explanation, which is that a congruency in one even very complex trait is more likely and more simple, requires less evolutionary steps than you try to explain all the differences between them if you were to put them in the same group. All right? And then also you would have to explain why they're more similar to other things. Right? And so it becomes too complicated. So for parsimony and likelihood, you don't put them like that. I also like to point out, since we're talking about cladograms again, that we use traits. Remember that if you did Whipple evolution, the whole point of that is to see that you can use both physical traits, niche, and DNA to make fine determinations on where to put people in the tree of life. But nowadays we use phylogeny, which is DNA, right? Which is really what part B is all about. So, but when you do phylogeny, sometimes you get this gene which confuses you because it's most closely to the fruit fly. Are you going to contradict all the other evidence that says it's more closer? Are you with me? So it's more parsimonious, more simple, more likely, right? That is just not. It's a freak event that is similar to the fruit fly. Some interesting hypotheses that were proposed is someone said maybe there was some contamination in the sample because a fruit fly was preserved at the same time in the same place. So maybe that gene was not even from the thing in the first place. If you read the pre-lab, though, it says that they got that from the soft tissue of the actual thing. So that doesn't mean it makes sense. It's a nice try, but it doesn't make sense. What actually makes sense is that the writers of this lab, crazily so, decided to put a gene that didn't make sense to kind of try to teach you that lesson. That sometimes that happens in real biology, and then what do we do? We got to think about what makes the most sense. You know what I'm saying? Now, most people, because they thought two of the genes say bird, one of the genes say alligator, and this other gene just tells something that doesn't make any sense, I'm going to put it with the birds. But then you also have to look at what those bird genes do and whether or not they're highly conserved, right? And if you look at those genes, you're going to see reptiles on them as well, like alligators and things like that. It's just that the birds had a higher E score or a lower E score, right? So they showed up first. But there were also reptiles with significant E scores. So that means you could also make the argument that even with respect to collagen and ubiquitin, it is more similar to reptiles too. So because of that, because reptiles show up in all three of those genes, this is what I want you to understand. Those H scores on the top of the perplexed, whatever is less than 10 to the negative 4 is basically homologous to that. Yes, sure, birds showed up first in the list, but anything that's less than 10 to the negative 4 is considered highly homologous. So that whereas reptiles in highly homologous in the collagen and ubiquitin as well as birds. But on the mitochondria one, there was less birds and more reptiles. And the mitochondria is a faster ticking clock. So you should have put it closer to the reptile branch. That's the actual answer. So So it should have been something like, not in the alligator branch, but like something like in between the the branch that goes towards birds and the branch that goes towards alligators. So if you have like the alligator here and the branch that goes towards dinosaurs and birds, right? Maybe an offshot that. Are you with me? So not in the alligator branch or in the bird branch, but before you get to the birds. You know what I'm saying? So that's the most likely what you could most probably substantiate with that. I also want to point out, since we're talking about labs, that you guys are being kind of dumb here about something. Okay? All labs have extra credit labs. And that, in case you haven't noticed. For example, the lab 13 has 5 different labs that I threw in there that you can do for extra credit. All labs have virtual labs that you can do. And the virtual lab has a virtual lab platform. You have to download it, extract the zip file. All right? You can't just click on things. It won't work. You have to download it to your computer, extract the zip file, and run it. And then you can do the virtual lab. Right? There's a worksheet you collect the data on, you turn it in. That's how you, basically it's just doing the lab here. But unlike the labs here, they work. They always work. That's why I like the virtual labs, because you know what I'm saying? Because you get to see what it was going to be if it works. And it also gives you a general idea of what you're supposed to do. Now, some of the virtual labs are not exactly the same lab we're doing, right? But some of them are. So you be smart about it and think about that. You have a chance of doing a lab that works, that always is going to work, right? So, for example, for Part B, with the data Part B is due, you could have also turned in an additional extra credit cladogram activity, right? And there are more which are due by the time the final report is due, when my Part C is due, all right, for this lab, which is look at the calendar and do not procrastinate it. Work on it little by little. It's something that actually it's part of both labs 13 and lab 3, right? So they kind of like work together in that sense, because you're like researching an enzyme and talking about it, but you're also discussing the evolutionary significance of it, and then you're also blasting it, you know? So if you look at it, there's a link between lab 13's part C, D, and then part C of lab 3. They are kind of related. You're supposed to do it on the same enzyme. Understand? On the main AP investigative folder, where all the investigations are, there's a file there, Excel spreadsheet, where you're supposed to go in there and type in what enzyme you want to do or what topic you want to pick. And then once I approve your topic, I'll put a date next to it saying, okay, I approved it. And I'll probably email you and say, your thing. If you make a modification to that sheet, right-click and share with me, right? And then once I make a modification and approve it, I'll give you a reply to your email and say, here it is. But that's where you select the enzyme you're going to do. You're not really supposed to repeat what other people are doing, right? That's why it's there. And sometimes you have explorations of your own throughout the year, things you're going to look at yourself. You're going to make decisions. For example, Part E, right? It's not actually one of those things, because everybody's doing the same thing with Part E, but you just have to develop your lab 4, you have to develop your own design. By the way, it's not, you don't get class time to do that. If you want to do it here, you can do it, but it's supposed to be homework and lab. But if you don't have a scale to do at home, you can bring it here, your potatoes, and do it. It's an individual assignment. It is not a group effort, all right? And you can do it here. You don't have to do Part E though, right? You can do the extra credit stuff instead, right? Or you cannot do it, because you only have to do half the stuff. Are you with me? But it's up to you how you take that. Now, you're not going to get class time for Part E. And you're also not going to get an additional time for do Part D and C, which you did not do. So then if you want to do those, you got to come in your own time. And that's why they gave me a planning period every day after school. Because it's impossible with the schedule we have to cover all the content we have to cover and do all the labs all the time that we have to do. It's not possible. Understand? So that's why it's like, for example, today, I got to use all these announcements and I still got to teach 3 case. Again, not possible. Which is why, if I teach my heart out of here, by the way, help me out there. There's like a rumor that I don't teach. That's crazy, right? Of course I do. I tell you guys to flip, right? To watch videos and to take notes and stuff like that. But it doesn't mean that I don't cover things over here. I always cover things over here. In fact, if you're preparing for the FRQs, it's what I say that shows up in the FRQs. But then the test also hits things that don't say, like any other college class. That's why you have to read the book. You have to watch Boltzmann. You have to use the review books to study. And you have to keep up with that homework. That is more important than the labs. It's more important than doing actual homework. Anything else, studying the book is the most important thing you can do in this class. I've told you this many times. So if you're not doing it, forget everything else but study the book. I'm serious. Right? Remember, if you got past the class with a C, you get a C no matter what. Right? And it's harder to get the same thing with work, because to do enough work to get a C with the work, it's harder. So it's almost smarter to study harder than to work harder. Does that make sense what I'm saying? Now, about the work. A lot of you guys are not doing EKs, you're not doing notes, you're not doing, like, for example, class notes. When I'm talking, you can take class notes and upload it to extra notes folder. Why aren't you doing that? Right? It's like a free grade. Everything I say, just write it down, upload it, free grade. Take advantage of that. All right? The quizzes, they give you a sample of test questions. Why wouldn't you do that? Always do the quizzes. So there's one this week for photosynthesis, right? So yeah. And then there's the EKs are designed to help you practice the kind of integration of information that you're going to need for the FRQs on the real test. So try to do the homework. But I understand if you're overwhelmed, you can't do it. That's why I said it's an hour a day every day in the beginning of the year. First time I saw you, before I even saw you, on the summer packet, I said that. Okay? Now, you're going to get a lot out of it. Those of you who already did the labs, you're starting to get the gist of it. It takes effort to do science right. And even if you try and you come prepared, shit happens when you're doing it. I saw it in the eyes of a few of you guys when you were doing it, how overwhelmed and how unskilled you were at actually doing it. Are you with me? How are you going to get better at doing science? By doing it, right? And that's why we do it. There's so much that you're getting from this class. Like literally. Let's talk about how to really make that lab time be more effective. Okay? So everybody clear on the lab 3 and what you really should have done? Now for part C of lab 3, you're going to get an enzyme and you're going to do, it's the hardest part. It's the part that you're really supposed to do, where you're supposed to create your own investigation and answer your own questions using BLAST. That is the character of the class. Part E is what you're really supposed to do. It's like it's the final part of it. Like, so that's what you're going to get to. But anyways, handwritten versus typed. Unless it says otherwise, everything is typed. Except for when you're writing the objectives, the summary of the background, materials and procedures, and your design that you're writing now, if it's a lab that you're designing yourself. Is that clear? So it's like this. If you were in college, you would have a lab notebook. Now, I'm not doing a notebook because we have this virtual portfolio thing, right? But you need to have, for anything that is the summary of the background, the objectives, which, by the way, is not just listing the objectives. It says to reflect on which one you know the least about and write about that, right? Then you're supposed to do materials and procedures for the lab you're going to do. And if it is a lab you're designing, that's basically materials and procedures, but you also have to have predictions. And sometimes I give you an actual scaffold of what you need. Like this lab that you guys didn't have already, it was all written there, the things you needed to have. So read the instructions carefully, okay? Everything else is typed unless otherwise stated. Is that clear? All right. All right, hold on. Pre-lab steps. The pre-lab steps, there's several things. The pre-lab prep. What I call the pre-lab prep, that's the reading the background, right? Doing the summary on it, and writing the objectives. You're going to prep step for that, grade for that. Understand? Then you also sometimes get a grade for materials and procedures type. Now, how does that work? The day you do the lab, you have to have that stamped and uploaded that same day in order to get a grade for that grade. When you look at the lab, you'll say the prep. That's just the background summary and are you with me? Now, I don't have any way to grade it, but you are supposed to do something else, though. It's not just summarizing the background and reading the objectives. Every lab has a shitload of prep steps. As in, like, watch these videos or read over this document. Are you following what I'm trying to say? To get you on board to what you need to do. For example, for lab 13, there's a bunch of videos. For lab 4, like we were talking about a video that shows what the LOD is supposed to look like on part C. And then I went home and I realized, hold on a second, I already gave them that video. It's part of the prep. Why is he asking me for it? Well, which means you're not doing the prep, even on one of the best of us. So make sure those things are there for your benefit. Now let me tell you something else. Are you going to have to do a lab on the AP test? Not really. You're going to have to analyze labs and write about labs, but you're not going to have to do a lab. If you were in Cambridge biology, they actually have your that. There's a day that you take a test, and a day that you do a practical final, that you do a lab. All right? It is awesome. In a way, I like that better. But here in AP Biology, you don't have to do that. You just have to interpret it. So deep down, what really matters and what a lot of AP teachers do because of it, because all they won't care about is passing the test, all we really need to do is sit here and talk about the labs. We don't have to do them, actually. We just need to understand them. Are you with me what I'm saying? But then what are we getting out of the class? That whole practice of the scientific skill, which is was a pillar of what you need to learn here. And if you were in college biology, you would have to do that for 3 hours a day every week. Are you with me? So that means we have to have 3 hours of lecture every week and 3 hours of lab time every week, and then you have to read 100 pages every week. That's how it is in college. Good colleges. I'm not talking about, some dual enrollment classes. By the way, if you're taking Introduction to Biology dual enrollment, you're not taking AP Biology. All right? You're taking 1 out of 3 classes, which actually is what AP Biology is like. Are you with me there? So this is a lot more than that. Right? It's designed to be harder. They cut the course, but it's still hard. And you need to understand that. You need to internalize. You're learning a lot. Okay? Now, like one thing I want to say also about learning. When I teach here, it's passive learning. You're not learning anything by sitting there listening to me. Not much anyways. Usually it's enough to get people to pass the test with the 3. It happens every year. They listen to me, don't do anything in the class, they pass. But it's only when you go home and you review what I've talked about in class that you truly learn it. You got to teach it to yourself. Are you following what I'm trying to say? And that's showing you the FRQs from a lot of you guys that you're not prepared because you're not going over what we talked about in class. Right? And it's not just reading Campbell. It's studying Campbell. Watching Bozeman, watching Crash Course, watching my videos, something. Going to the videos in my tutorials folder, looking at stuff, doing whatever you need to really understand this. What I'm going to do today, trying to cover the Krebs, the citric acid cycle, cell aspiration, photosynthesis, all in less than an hour, it's crazy. It's impossible for you to truly understand it. So those of you who didn't read it are going to be totally, you've really lost. However, there is a video in my tutorial that walks you through it awesomely. By the way, I just told you about animations in biology, bioflicks. They're usually awesome. There's a bioflicks on cell aspiration. There's a bioflicks on photosynthesis that breaks down each stage. And there's a series of animations in the videos at the tutorials folder. If you are lost in these 2 topics, make sure you watch those animations. On each topic, this is under cytology, and then it goes into cell energetics, and then you can do the videos and my tutorials there. Trust me, they're awesome. I'm going to try to find a time today to show you a piece of one of them so you can see why it's so awesome. And it's really worth looking at it. And the internet is full of resources. You know how I put together all these resources? Professors at Northwestern, at Harvard, at Stanford. They have websites too, and they do the same thing. So if you're desperate, just freaking Google it, but do something. You cannot walk out not understanding these cycles, these steps of both of them. I understand. Okay, I'm going to do the best I can. Continue that. Make sure then you're doing the pre-lab steps. Don't just do the summary of the background and write the objectives down. Actually prepare for the lab by doing the pre-lab steps. The pre-lab questions. 30 to 40 questions. You saw those? I used to give those as FRQs. Back in the day when biology used to be factual. So I don't have those anymore. I just put them over there. Now, I give them grade for almost completion. I scan through them and see if you're doing it right. Sometimes I see, no, not doing so hot. If you're answering those questions and you don't know how to answer it, or you think you have it wrong, make sure you ask Ms. Lima, put it on the student file, you know what I'm saying? Ms. Lima, this is a question I did not know how to answer on the pre-lab. However, everything is graded separate. So the prep is a grade, materials and procedures is a grade. By the way, how do you get that grade? By the same day you did the lab, you have to upload it online, right? Now, the pre-lab questions are typed. And you have to do that by the day the pre-lab is due. So for example, for lab 13, it was supposed to be due yesterday, but now that I postponed it to Wednesday, all right, not to Wednesday, to Thursday, we're going to do the pre-lab is due on Wednesday night. But you already have homework that night, so, if you did it, great for you. But Wednesday night, if you don't have the pre-lab uploaded, the questions there, send it to me, share it to me, it goes into the late folder, and then it might not get graded. Are you with me? Those are how the pre-lab questions work, all right? As far as the materials and procedures, the day you do the lab is going to be stamped and uploaded. Does that make sense? All right? Same thing for, so then, materials, procedures, predictions, and design is due the day you do the lab, upload it. Lab notes. The day you do the lab, if you want the lab notes grade, you have to have a very organized data collection protocol, right, that you did in class. You have a chart. And as you walk out of the room, you stamp it. And then you take it home and scan it and upload it that same day. Otherwise, you forfeit that grade. Now, you only need half the grades, right? So it's okay if you didn't do it. It is not okay for one person to have the data, send it to everybody, they do their own sheet based on that and upload it. It's not going to count because you need a stamp that same day. Is that clear? And this is not a group effort, it's individual, the lab notes. Okay? So, by the way, if you didn't do it for the previous ones, do it for the future ones, but make sure that you have it ready and leave the classroom with that data. Now, I had to go to class and it wasn't done yet. That means before you leave, meet up with that person and then get a copy of it, come back over here, get it stamped. That's the only way you can get that grade if you really want it. Do you understand that? Or you can come super prepared for the lab, you know exactly what you're doing, everybody has a job, and you get it done in the hour and a half that you have to do it. Are you with me? And they don't have that problem. Okay. And then the report. Report is due 1 day after the lab is done. Okay? Each part's report. Now, here's how it works. There is a folder on the AP Investigative Labs that's not a lab. It's like it goes to 1 to 13, and then the next folder that just says Lab Report Writing. And in there, there's materials that give you instructions on how to write your lab report. Understand? Now, here's what happens. And this is actually the first one we're doing this for, because the real first lab report is Part A, which was due on Friday. And now, Part B is actually due, tomorrow. That's today, because it's a week after Part B. Now, which means why? You could have done Part B late, but if you don't do it by the day the report is due, you won't be able to do the report grade. Does that make sense? Okay? So you can get a late, by the way, anybody that does it gets a grade for doing it too. So everybody last Friday got grades for Part B, right? And some of you guys got grades for Part B the first day. Are you with me? So you get grades for doing it, just for doing it. Unless you do something bad or not, if you're clearly like slacking off, then I don't give you the grade. Or, it's clear that you don't come prepared. Now, I'm going to be more stringent as time goes by because you're getting used to doing the labs. But at the time in January, when you're doing a lab, I expect you to have performance. You're doing the lab well. Understand what I'm saying? Because it's a learning curve. Now, the last thing I want to say about that is that then, if, for example, if you haven't done Part B, everybody has done Part B, right? Okay, but if you hadn't done Part B, if you could have come today, for example, done it today, you could even have come next class and done Part B next class, but you could never get the lab report grade, or you could, but it would go in the late folder and it might not grade it. You know what I'm trying to say? Because it's due today. Is that serious? What I'm trying to say? So if you don't submit it today, there's a chance you can get graded, but there's a chance you why not, because it will go in my late folder. Does that make sense? Okay. About that, for Part B's report and Part A's report. When you send me your Part A, I will forward someone's Part A to you. Okay? This is going to happen from now on. And you have to do peer review on the other person's work. What that means is that you are going to be the person who's receiving the paper. This is how it actually happens in science. Okay? When I submit a lab to a journal, somebody else, completely different place in the world, gets the paper, goes over it, and see if it makes sense. Are you with me? So if you want to be a scientist in modern society, that's kind of how it works. Your paper gets reviewed by others, you review other people's papers. Understand? And you learn both ways, because by seeing what they did, you say, oh, okay. That's how you do a lab report sometimes when you did a bad one. If I'm trying to say, maybe next time you do a better one. And I will also choose the best Part A report and share with all of you guys so that you can see how it's supposed to be done. You know what I'm saying? For those that did it. Now, the way you do it is like a rubric. There's a rubric to grade the lab report, and that's on the lab report writing folder. You are supposed to get that rubric and then reply to the person. You know how when you share something with someone on Drive, you can actually leave comments. I have done it for some of you guys. You can insert a comment. So you can just insert a comment, okay, so you just lost point H1 because you did not have the hypothesis like this. If I'm trying to say, there's a rubric that you're supposed to follow. Are you with me? And you're going to give the person feedback on how good her report was. Does that make sense? I will also go over the report and give the person feedback. Are you with me? That has nothing to do. I'm the one that's grading it. It's just that you also get a grade for doing the peer review. Understand? Now, in order to get a full credit for your lab grade, you have to do the peer review. Otherwise, you don't get full credit. That's kind of how it works. So it's like 3 grades for the report for part A, for example. All right? You don't get the 3 grades if you don't complete somebody else's peer review. Does that make sense? You get 2 grades instead of 3. Does that make sense? All right? So stay tuned for that. You'll receive the peer review thing. Don't go crazy with it. Just open up the rubric, read the person's thing. Oh, this is missing. It's not in there. Oh, this is missing. It's not in there. Are you clear? All right? All right? And it's like you're not really giving the person a grade. I'm the one giving the person a grade. And I'm very generous with the report. If you got a decent report going, you get the 2 grades. All right? It's more so that you know how to write better next time. Understand what I'm saying? Okay? All right. So that was the announcement I had to do about the lab. So let's go back to 2A.1 and talk about it. So those of you who are smart are taking notes. When we talked about 2A.1 last time, so can I have the lights, please? When we talked about 2A.1 last time, we talked about the types of reactions, endergonic, exergonic, right? So, and we talked about one of them releases energy, the other one absorbs energy. Or one of them releases heat, the other one absorbs heat. One of them increases entropy, the other one decreases entropy. One of them increases enthalpy, the other one decreases enthalpy. One of them adds free energy, the other one takes free energy, right? One of them raises the temperature, the other one drops the temperature. One's spontaneous, the other is not spontaneous. Are you with me? And it's also important to know examples of things that release energy. For example, cell respiration, digestion, right? Hydrolysis, ATP hydrolysis. Things that consume energy, right? Which talk about building ATP, photosynthesis, all right? Protein synthesis, cell replication. Most of the things that life needs to do, we talked about this in the lecture, to stay alive are anabolic. Energy consuming, not spontaneous. And for life to do that, it has to pair those reactions with exergonic reactions that give out energy. But because, we talked about this, most of the energy goes to heat. So when I destroy something, I can get the energy out of that, right? When I break the bonds. But since most of the energy goes to heat, I have to break down a lot more than I build. So since life, in order to maintain order, is a constant drive to build, does that make sense? You actually need to destroy a lot more. So is life a destructive power or constructive power? Destructive, because it destroys a lot more than it builds. So all the advancement and evolution and organization that you've done in your life and that we have done as a species in the history of life, of Earth, right? All of that has been done at a vast expense of the energy from the environment, mainly the sun's energy. But, and it's not like as if it was going to be wasted anyways, right? The sun is just radiating the shit. So we just decided to grab some, right? Does that make sense? Right? But you have to understand that, though. It's a very important concept. Entropy always increases. So life, when you became more organized from one single cell to the 6 billions you have today, and you're still fighting entropy every day, and for that you have to build a lot, spend a lot of energy. All of these things you're doing, all right, are doing at the expense of even more destruction. Think of all the food you destroy, all the trash you create. So life actually destroys a lot more than it builds. The one thing I did not talk about as far as metabolism is concerned is the role of enzymes in this. Now, if a reaction is like this, which of course we are at a higher level of energy in the reactants than we are at the products, which means energy is being released, right? Which means enthalpy is going down, which means free energy is increasing. We're increasing disorder. We're increasing the temperature. This is going to be spontaneous, right? Cell respiration, digestion, hydrolysis, examples of this. But enzymes act to kind of cut this activation energy and make it lower so that I don't need as much energy to start it. You need less of a spark to get the thing going. Notice that the final and the beginning do not change. So the enzyme's job does not change the entropy change. Does that make sense? It does not change the enthalpy. It does not change delta G. It's all the same. Does that make sense? So what does the enzyme do really? Only lower the activation energy. All that does is it makes things go faster. Right? It's a catalyst. What does that mean though? How does the enzymes do that? So the enzymes have an active site, right? that fits exactly on the substrate. The substrate is the thing the enzyme is supposed to work on, correct? Now, some enzymes break things down, like this one that's doing this job is a breaking down enzyme. And then what basically it does is that in that active site, it creates the perfect environment for that reaction to take place, right? Which makes it easier for the bonds to be broken and therefore lowers the activation energy. Does that make sense? So there are other enzymes which build things. So enzymes that help this reaction, all right? Now this reaction, you have more enthalpy at the end. You're going to go down in delta G. All right, you have temperature is going to go down. Entropy is going down because you're getting building blocks and building more complex things. Photosynthesis, protein synthesis, cell division. And half of our life wants to do is like this, right? More life wants to do this a lot. In this case, the enzyme, there's no activation energy, so to speak. But the enzyme can still make this go faster. How does that mean? Because if I say I want to put this and this together, they will have a particular way to go together. Think of it. They don't go together like this necessarily, like this. They have a specific way to go together. Are you with me? So they have to go together. So let's say putting this together. I can't put it like that, or it has to go exactly like that. So what the active site does in this case is create the perfect shape for this thing to match just right, and for the other thing to match just right, bringing them close to each other exactly in the alignment that they have to be. Are you with me? Which makes it easier for that bond to take place. Does that make sense? And so enzymes speed up the chemical reaction by creating the perfect environment for the bond to form. Here they create a perfect environment for bonds to break. Here they create the perfect environment for bonds to form. Does that make sense? Right? And that's how enzymes facilitate the chemical reaction. But enzymes are proteins. So if you mess with the temperature, what happens to the enzyme? It denatures it and it stops doing what it does. So pH will interfere with the bonds that exist in the, by the way, don't forget to do that macromolecules chart. Next time we have a chance to do that for Q, you're going to want it, right? Right? So anyways, enzymes have those, are held together, the quaternary structure of enzymes is held together by hydrogen bonds, disulfide bridges, electrostatic interactions between charged particles, hydrophobic interactions, hydrophilic interactions. That's what holds together that tertiary quaternary structure of the protein. You're going to break that down, though, if you start playing with the pH, because it interferes with the formation of interactions between the positive and negative charges, the hydrogen bonds, and things like that. So that will cause the nature to elucidate, and will also denature the enzyme just the same. Once you denature the enzyme, there's no way back. Now you got to destroy the whole thing and rebuild it forward. Just like the egg. Once you fry it, there's no going backwards. You can get back to the albumin it was before. Does that make sense? So that means temperature, pH, can have influence in the enzyme activity. We already talked about this, but if I have to find a single piece of Lego in a bucket of 2,000 Legos, and I'm the enzyme trying to do that, and the Lego is the building block that I need to work on, that makes a lot of job a lot harder. So when concentration of the substrate is very low, the enzyme also slows down. Does that make sense? So for example, if you do a graph of enzyme activity, all right, and you talk about, so this is what the enzyme activity is, what this spawns. That goes to the dependent variable. And I'm going to change a few things. So think about pH. If I'm doing that, there's going to be an optimal pH that the enzyme is supposed to work on. Any deviation from that is going to make it bad. So how does that look like? How does that look like? What does it show like? No, not a straight line. There's an optimal pH, and any deviation from that makes it worse. So how does that look like? Right? Something like that. Very good. A lot of people drew it. Right? So that's good. The same thing would be for temperature, because it has to do with that denaturing thing. Now, this is interesting with temperature. If you heat it up a little bit, it actually helps. But not because it's helping the enzyme. It's just making the molecules move faster. So it makes more collisions, it makes it more likely for the reaction to take place. But if you heat it up too much, you have a problem. So the temperature one actually looks like this. You increase the temperature, actually helps, but then it gets to a point that denatures, so it has a sharper line after that. Are you with me? Now, what about if you're talking about concentration of the substrate? Well, the more substrate you have, the faster it goes, right? Up to a certain point, though, because at a certain point, it doesn't matter if you have 1,000,000 more substrates, there's a limitation from how many enzymes are there to work on it. Are you with me? So that means, remember I told you about this, anytime in biology that you have a limiting factor, what happens to the graph? A flat line. So how does that going to look like? It goes faster and faster and faster up to a certain point at a flat line. At a certain point, adding more substrate doesn't make a difference because the enzyme is limited by how many enzymes are there. Which means if I added more enzymes, I could make it go faster. Does that make sense? Because it's a limiting factor, right? Now, if I ask enzyme activity over time now. So let's think about that. Let's say I start with a bunch of substrate. This is what you're supposed to do in one of the labs. You start with a bunch of substrate, right? What's going to happen to the substrate over time? Well, remember, enzyme activity over substrate concentration, right? As you have a lot of it, it goes really fast. But you have a little of it, it goes very slow. Remember that. So at the beginning, you have a lot of substrate. So it means it's going very fast. So I mean, what's happening to the substrate? It's going what? Down very fast, right? Talk about, so this is enzyme activity. Let me graph substrate concentration over time. So I start with a lot. But very quickly it starts to drop because the enzymes are working very fast because a lot of them. But as they start becoming rarer and rarer, what starts happening to the rate? Slow down, right? And so that means it's going to slow down and down and down until there's barely any substrate, and it becomes very hard for the enzyme to fight it. If you're graphing enzyme activity over time instead, right? What is that going to look like? It's going to look like this, right? You start at a constant rate, which depends on how many substrate you have. It's limited by the enzyme, so it's a constant rate. But then as the substrate starts becoming more rare, what happens to the rate at which enzymes act? It's going to slow down, right? And eventually flat line because of that. So this part of the graph right here gives you the rate of the enzymatic reaction. That part right there, it's just because you're running out of substrate, so it doesn't go any faster than that. Does that make sense? All right? So these are some graphs that you're going to see in tests, and I just went over the pattern there, all right, on all of that. Okay, now I want you to understand, too, something else about enzymes then. Enzymes prefer to work in situations where they don't have to keep looking for their thing. We already talked about that. That's why compartmentalization makes sense in the cell, because you have the enzymes working in a place where they don't have to look so hard to find what they have to work on. All right? The contrary of that is called something called competitive inhibition, because basically the enzymes have to find the substrate and a bunch of other ones. Our special type of competitive inhibition is when the active site of the enzyme itself can become blocked by something that has a shape very similar to that of the substrate. This is actually how we shut down some bacteria, some viruses, some stuff, because we use a drug that inhibits the action of something that's causing a problem in your cell, in the body. Are you with me? So some drugs actually work that way, by inhibiting the action of enzyme, by mimicking the actual substrate. All right? It's called an antagonist. It's very good. All right, antagonist. Now, there's another way of doing it too. Some enzymes have an on and off switch, right? A secondary place on them, where if you put something on there, it would change the active site shape, and then it can no longer fail the substrate. Or it's the other way around, where they only open the active site if something goes in there, and then it opens the active site. So in some cases, it's like you need to have the key to make it work. In other cases, if the key is there, it shuts it down. Either way, the body can use that to regulate whether or not the enzyme turns on. But since enzymes are necessary for things to go faster, that's actually a way to regulate the body's functions altogether. Are you with me? And this is called allosteric activation and inhibition. For example, in cell respiration, which we're supposed to still talk about somehow miraculously today. All right? In cell respiration, right? Think about it. If I don't have any oxygen, I shouldn't waste time going to the mitochondria and producing a bunch of these electron carriers, which I can't use on the ETC because there's no oxygen to run the ETC. Right? And so the body shuts down the mitochondrial processes in the absence of oxygen, so that you don't waste time producing all these electron carriers which the body cannot use unless there's oxygen. So it means there's an enzyme somewhere along the pathway who shuts down in the absence of oxygen. So you can think of oxygen, right, as something that does what? Allosteric inhibition or activation. In this case, it would be activation. You have oxygen, the enzyme works, which means the process goes forward. Does that make sense? Likewise, does it make sense for us to make a lot of ATP if there's already a lot of ATP in the cell? No. So it means ATP is the final product of cell respiration, correct? The actual final product, ATP, inhibits the enzymes that work on breaking down glucose in the beginning of it. You understand what I'm trying to say? So you have, at the beginning of the cycle, you have an enzyme that works on breaking down that glucose molecule, which I consider like the gold bar of energy, right? That enzyme has a little allosteric site. At the end of all of this, you're going to make a bunch of ATP. That ATP is actually what shuts that down. Are you with me? That's a classic example of negative feedback. All right? Because the more of this you have, the less of that. Are you with me? So it's a feedback loop where you're turning off to stop something that's happening. And enzymes, therefore, are not just catalysts. What are they really? In addition to being catalysts. What other job are they really doing here? No. The enzyme is not just acting like something that speeds out the process. By the way, what else is it doing? Yes?
Like population control.
Well, not population, not talking about population, but that's the right idea. Yes? Sorry, what did you say? Prevents, what is it doing then to the reaction? It's what? Regulators. Right? So it's a regulator too. Does that make sense? Okay. So that's all important stuff that I have to talk about. Now, going back to 2.A.1, there's some stuff completely unrelated to this whole. This is a cool thing about what they did with this new curriculum, is that they're not talking about chapters. They're actually talking about concepts. So this concept of 2.A.1 is actually about energy, right? Energy in life and how life requires a constant input of energy. But there's two ways of tackling this problem. Destroy more to get more energy, because remember, if I need one little block of energy, I probably need to destroy something that can give me 10, because 90% of it is going to go to heat, right? But that means destroying a lot, because I need a lot of blocks. So think about it in terms of economy, right? If you need a lot of money to get your business going, there's two ways you can do. You can generate more cash flow, or what could you do? No? Steal, I'm sure. Life does that too. It steals. We'll talk about that in a second. But what else could you do? What do companies do to stay afloat on the times of crisis? Right? Not only can you generate more cash flow, what can you do? Right? Which is, what are you trying to do when you're removing the employees? You're trying to do what, really? Save money. So life can do two strategies. I increase the input of energy, or I also decrease the need of it. Does that make sense? How do companies do that? Sometimes I don't want to fire anybody. What do companies do? They look for more efficient ways to do things. Right? Or they even pair up with another company. You see where I'm going with this? So the life forms, evolution, because there's a pressure to do so, a long time ago, any mutation that allowed this kind of stuff to happen was selected for. So this process of saving energy is a characteristic of life that has to do with this 2.A.1 topic. So let's talk about 2.A.1. All right, and how it fits with this idea. By the way, very useful tool to have on Google Docs. Things that you use all the time, share on stars, because you're going to use it all the time, right? So essential knowledge I just want to make sure that I don't forget what I need to talk about. And then I'm going to go to the other board probably because I need to write. So we're doing 2.A.1. Yes, go ahead. So where we are, by the way, on 2.A.1, they also list specific processes which are associated with this idea of input and output of energy. So for example, we have the Krebs cycle, glycolysis, Calvin cycle, and fermentation. So let's think about it. The Krebs cycle, which is also called the citric acid cycle, shows up in cell respiration to break down molecules and make ATP out of it, and all these electron carriers, which ultimately get used in the ETC to make energy for the cell, right? So this is an exergonic process then, one of those that give out energy. Okay, so we'll talk about Krebs, and then we've got to do that. We got to sometime we're going to talk about it today, but that is one of the exergonic ones, right? Exergonic. Glycolysis, is that also releasing energy? Certainly. So glycolysis also belongs here. Glycolysis. Then we have Calvin cycle. The Calvin cycle requires an input of energy from the light reaction of photosynthesis in order to power the capture of carbon dioxide to build G3P molecules, which are going to be used to build macromolecules, right? So that is the intaking energy. So that's actually endergonic. Endergonic. And it's like the inverse of the Krebs. So this one is citric acid cycle, which is basically destroying acetyl-CoA. Well, it starts with oxaloacetate. We'll talk about that in a second. And it's a cycle. They're destroying molecules to grab energy out of them. Calvin's cycle, in the other hand, is about using building blocks and energy. Right? So it's endogoning. And we have fermentation. That's also about generating or using energy. So that's going to be here also. Fermentation. Okay? So energy-related pathways in biological systems. Now, I added to it when I talked about that there are more over here that's like, you can talk about in terms of the ETC, you can talk about protein synthesis, ATP hydrolysis, but these are the ones that they specifically mentioned. On this bottom part here, we actually already talked about this part, which is you can apply this concept of needing energy to the population level, to the organism level, to even to the ecosystem level. At the organism level, this is what this is all about. We're going to talk about it in a second. But at the population level, the more energy there is, the more the population grows, right? When there's a limiting factor, the population flatlines, right? It usually is, say, not enough productivity from the producers or a lot of predation, right? There's a lot of these limiting factors. That could be a disease. It could be a lack of nutrients, lack of sunlight, lack of water. Whatever the limiting factor is going to prevent the population from growing. So both matter and energy, but this is specifically talking about energy, so it's really talking about productivity. If the producers are not capturing enough sunlight and producing enough sugars and macromolecules for the rest of the food chain, the populations will actually start crashing because there's less energy available. You can actually apply that to the ecosystem level in the idea that the more productivity there is, the richer the ecosystem is, like the rainforest, for example. Why is it so diverse? Why is it so rich? Why is there so many life forms? There's so many types of life forms. What does that say about the productivity of the rainforest? It's got to be what? Really high. Why is the coral reef so diverse? High productivity. Why are grasslands so diverse? A lot of productivity. Why is the shorelines so diverse? A lot of productivity. See what I'm trying to say? So that means the more productivity you have, the more resource you have for the trophic levels to become bigger. And you become bigger, you're expanding niches. If you're expanding niches, you can have more diversity in those niches. Are you with me? So that's why this concept of need, you see, the more energy you have, the more order you can get. And that can be applied. This is a concept that's overlying. It's not just about a cell. It's not about an organism. It can be even a population or even the ecosystem as a whole. The more productivity you have, the better. The only caveat I have about that they don't go into, but I will say, sometimes vast increase in productivity actually increases competition, which actually decreases the diversity. If it's very sudden, I talked about that very quickly. And sometimes things like eutrophication, when it's like hyperproductivity because of too many nutrients, actually ends up killing the lake because it blocks the sunlight for the life that's underneath it. And then actually the algae starts to die, and then all the dead algae goes to the bottom of the lake, gets decomposed, it actually produces a massive amount of carbon dioxide, and actually suffocates all the life below the carpet of algae that's on the top. So it's actually ironic that you're increasing productivity, but causing the destruction of the ecosystem because of it. So this is a caveat. So there is certainly a pattern that more productivity means more, right? But sudden increases in productivity could be chaotic, both because of increased competition and because, in the case of eutrophication, you're getting too many producers, almost suffocating the rest of the life. Does that make sense? All right. But look at for example, on the ocean. In the water, coastal things, the trophic levels are weird. There's actually less producers than the rest of the levels, right? And that's because they regenerate so fast that they can't eat them fast enough. Are you with me? So there are always less phytoplankton than there is zooplankton in coastal oceans, right? But because the phytoplankton reproduces incredibly fast, right? Which tells us something. It's not necessarily the biomass that matters to measure the productivity, right? It's the increase in biomass that matters. That's important. If I'm trying to say, so it's the change in biomass that shows you productivity. There's other ways of doing. By the way, if you look at the cell respiration photosynthesis formula, which since I'm talking about it today, should definitely be on the board. So let's put it in there. Should have word of short name, except I had the spirit thing. But if you put So these are things you're going to need to do photosynthesis, right? And these are the things you're going to get. Now notice there's ways to measure productivity, therefore. I can measure the amount of biomass increased by the sugar that was produced. Now remember, this is actually not what photosynthesis actually makes. Photosynthesis makes G3P. Now G3P can be used to make glucose, but it can also be used to make proteins. It can also be used to make nucleic acids. And it can also be used to make lipids. Where it goes is different. But it could do that. Now, the oxygen also gets produced. So what is another way it can use to measure productivity? What it can do? The way, if you watch Boltzmann's lab, that's what he does in his lab. He measures the amount of oxygen produced. And you can do that lab as an actual lab if you want to. We have what we need to do it. The LLD are right there. You put the LLD inside of a test tube, close it up completely, and then come back a few days later, you have a bunch of oxygen in there. Right? It actually starts creating a little atmosphere. You know? Because it's producing. You put it in a strong light. Right? Now, you could also measure what? If you want to try to figure out the productivity of point not just the biomass increase or the oxygen, what could you measure? The loss of water, or what? The loss of carbon dioxide. So satellite imaging can actually measure the concentration of carbon dioxide in the atmosphere. Right? And so over a forest, if you look at changes in carbon dioxide, you can actually measure productivity of the whole ecosystem. You could also do the same thing by measuring the amount of green increase, right? Which is called a standing crop, basically. How much is it actually increasing that thing? That's actually what we're doing. Once we killed the plants last week, we had mass. But it was wet mass. It was full of water in there. So you put it on the stove and it dries out. So you have this dry mass. And that's what you measure to figure out what's the actual biomass. Now if you compare it to week 2, right, and the week 3, there's actually been, and I didn't show you guys this, but towards the end of the lab, we're going to go over the data. There's actually a 50% increase this week. So the plants actually got, like if they were 10 centimeters, they got to 15. But not, remember, plants don't grow just up. They grow thicker. They go to the sides. Total, they go down, the roots, right? Total biomass increase by 50% of the body. Now imagine if that was you. It's like I'm at 6 feet, that's coming back next week and being 9 feet. Think about how fast the mitosis is happening in there. Right? These plants are growing very, very quickly. Right? So much quicker than we can grow. Right? So they're going through a lot of productivity so we can actually measure that and quantify the productivity of these plants. We can actually quantify how much carbon dioxide was used. Because there's a direct relationship between the amount of sugar produced and the amount of carbon dioxide produced. Right? So if we think about the biomass increase as sugar mass, and it is because it's mostly cellulose, right? Then you can actually create, and the lab talks about the formula you need to do this. In the pre-lab, for lab 10, you're supposed to read, there's a formula you have to use that you can relate the biomass directly to the amount of carbon dioxide consumed. And therefore the amount of oxygen produced. Right? So we can figure all that out. And it's very interesting. Okay, so the next thing here is about saving energy, right? About saving energy. So, for example, organisms use several strategies in order to regulate their body temperature. Why is it so important to regulate your body temperature? Now, go back to earlier today. Why is it so important to regulate body temperature? Enzymes. Okay, so then you need to regulate body temperature. Enzymes work at optimal conditions at a certain temperature. So if you're too cold, the enzymes start what? Now, if you got too hot, that's bad news because you're going to have to redo all the enzymes. You're going to have to spend energy to rebuild them. So, what is the organism's strategies? Now, the ones that are lucky enough to have evolved protocols to burn or use their, they literally burn. Yes, Enzo. Because you're trying to kill someone's enzymes. It's the bacteria's enzymes you want them to die. Now, you have 6 billion. You're hoping that they will die before all your 6 billion die. Are you with me what I'm saying? Not only that, though, you're also increasing the temperature, which makes everything happen faster. Right? Like the collisions and stuff. So it accelerates the immune response and up to a point. And then it starts killing the enzymes. But the problem is, of course, you don't need that because antibiotics can kill the bacteria. Right? So then we take medication to lower the body temperature because they say, okay, stop with this 2 million-year-old strategy to survival. We don't need it anymore. Are you with me? I'm saying this is what we're telling our body when we take the acetaminophen. Yes.
But say that was possibly like, even though it's using like an use exergonic or
We're going to talk about that
Back to what
at some point. Yes.
you think
Yes.
that evolution, evolution, whatever, like take that fevers out, so we don't really need it anymore.
If we keep doing it this way, sure. People that are born without fevers have a bigger advantage. But that will happen more in populations where fevers are more common. So you'll be expected to find that where? Where are people getting a lot of fevers? No, because then there's no pressure because to not have them, because they don't have the medicine necessarily. But I'm saying you have to be a place that gets a lot of fevers, but the medicine is very big. Are you with me? For that to happen. And it would have to take thousands of generations for that to occur. So that's going to take a while, sort of speaking. But I was talking more about a place that has endemic diseases that causes a lot of fevers very often. So tropical areas, right? So like where you're going to get a yellow fever, dengue fever, all those kinds of things are going to be more common, for those things. But, all right, endothermy. Endothermy means having a regulated process to burn glucose, not for energy, just for the sake of wasting it. Because when we do cell respiration, right, it's for ATP, right? Actually, that's not accurate for you and me. 80% of the reason you do cell respiration is to keep your body warm. All the other ATP is just a bonus to be getting out of it. Are you with me? We actually intend to waste the energy as heat to warm ourselves up. Are you with me? So it's not a waste at all. But that means we're going to constantly burn. What does that mean of our metabolism compared to the metabolism of other organisms? It's got to keep burning all the time, even when we're asleep. Are you with me? Now, because of diffusion rates, something with a larger surface area to volume ratio loses heat slower. So it needs to do that less. So a little mouse, right, which has a big surface area to volume ratio, because it's tiny, right? Remember, the tiny things have a bigger surface area to volume ratio. That means a little mouse loses heat quicker, which means it's burning glucose even more to keep its body temperature. The elephant, on the other hand, being very big, has to burn less sugar for that purpose, even though it's bigger, right? Now, comparatively, of course, it's burning more sugar. I'm talking about percentages. Are you with me? Percentage of the energy of the mouse that's spent towards keeping its temperature. The mouse is going to spend more of its energy doing that. Why? Because it loses heat to the environment more. Does that make sense? So being bigger actually allows you to conserve heat. But it also makes it harder for you to cool off. If you think about it. Does that make sense? All right? Which is why the large endotherms have mechanisms to cool themselves off. Like the elephant has a lot of blood vessels in their ears to kind of dissipate the heat. They're like radiators, literally. All right? And we have sweating, right? So the large endotherms do that. Ectothermy, on the other hand, is doing what? What do? To heat yourself up if you're an ectotherm. If you're not a mammal or a bird, what are you doing?
You're cold, so you have to use outside.
So if I want to get warmer, what am I going to do? I got to go to a warm place. And in that warm place, I'll get to act a lot. Or if it's getting too hot, what am I going to do? Go to the water or go to the shade. So that means they act as much as they are allowed to act based on the environmental conditions that are available. Which means during the winter, you don't see as many bugs. Why not? Because they don't have enough warmth to be moving around. So they're hiding somewhere in like hibernation. Does that make sense? All right? Okay. How about the same thing true about plants? Some plants actually regulate their temperature. Because in order for them to flower, they have to flower at a specific temperature. Does that make sense? Temperature is going to determine the flowering time, which is why global warming is changing the temperature of the flowering times of plants. But some plants don't want to wait for their optimal conditions. So they actually expend sugar just to warm themselves up in order for it either for the seed to sprout or for the flower to flower. So some plants actually have mechanisms of warming themselves up to do that. Whether it is a darker colored leaf or a darker colored flower to actually retain more of the heat. You know what I'm saying? Or it is actually a metabolic process that's actually making more glucose be burned. Some plants do that. So these are examples of regulation mechanisms designed to save energy, right? Or to spend energy. Why? Now it may seem crazy that we're burning sugar to save energy. But we are, because if we weren't, our enzymes would not work. And so it would take more energy to do everything. Does that make sense? Right? Now, of course, for the endotherms, they just go to the sun when they... But that means that they depend on the sun. If the sun doesn't come out, they have an off day. Does that make sense? So there's clearly the endothermy has an advantage in certain environments over ectothermy. Yes? Yes. They use external thermal energy to help regulate and maintain the body temperature. Right. I don't like cold-blooded. Use the right term, ectotherm. We're not in like middle school anymore. All right. Also, here's another smart thing to save energy. Why don't life forms all reproduce, have babies in the middle of winter? Why not? By the way, it's not, there are exceptions. But especially up north, where the winter is really harsh. Why are we having babies on the winter? You're going to use that energy to have the baby, and then as soon as they're born, you're going to have to feed them, take care of them, with what? The resources depleted during the winter time. Which means a very important strategy is to time what? Time reproduction cycle. Which is why the heat cycles of when they go into heat, right? Which ironically, because it's about saving energy, heat, right? Is time. And you see that in plants, and you see that in animals. Seasonal reproduction matching the environment, especially in places where the seasons are drastic. Florida, not so much, but in drastic season, does that make sense? You will also see life phase respect. There are some plants which are reproduce once. It's called itoparity. One reproductive event. You know why they do that? Because they don't know if they'll have another chance. So they evolve to take that one chance and make a billion seeds, pollen everywhere, to try to have that one chance. So they have explosive amounts of offspring to try to hope that one of them makes it. Even trees, there are trees that do that. Whole enormous tree. It waits for that one season, and then it blooms, and it will never bloom again.
So like they wait till like the perfect season.
Right. And then other organisms, on the other hand, don't. Now, so biennial plants, they do it twice per season. There's perennial plants that do it all the time. Right? So it depends on where they live, where they evolved, and what the condition of the environment was. Birds. When the food supply is limited, they have smaller flocks. Not flocks. The size of the brood is smaller. Are you with me? Now, you can see that here. There's like 55 ducklings when they have children because everybody feeds them. So they have a lot of food, and they feel, okay, so I can have a lot of babies. Now, if you're going to have, it's interesting though, if you think there's going to be another season, right? If it appears based on previous seasons that the seasons have been healthy and coming on a regular basis, then is it more advantageous to have an enormous flock or to have less children and make sure they all survive? Right. So when there is a pattern, you actually start decreasing the flock size. Right? If there's a sudden increase in productivity, a sudden increase in offspring. But if there's a pattern of constant productivity over many seasons, the brood size actually gets smaller. Because they think it's almost like instinctively or metabolically, because it's all controlled genetically, they know, right, genetically, the genes that control that respond to the environment. We learn about how this works later in the year to actually reduce the number of offspring. Are you with me? Because I have a better chance of each of them surviving if I have less of them. Are you with me? Because I can take care of all of them. And it's okay because the seasons have been good. That means the next season there's going to be a better season again, and I'll have another chance. So altogether, I'll have more children overall. Does that make sense? But if I'm an organism that only gets one chance, then I'll have a lot of children. Or if I'm an organism that hasn't had good chances lately because the seasons have been rough, then I'll go for more children. Does that make sense?
Yeah.
All right? So the point here is that there's a relationship between what the environment does and what happens because I'm trying to conserve energy. So that's the organism and the population ecology at the same time here. All right, no time for questions. I'm so sorry. All right? There is a relationship between metabolic rate. I kind of already talked about this. There's a relationship between the metabolic rate and the body mass, right? Because of the whole diffusion rate. right? So larger ones have a lower metabolic rate than smaller ones. This is among endotherms, obviously. You can't compare a bug and a mouse. Why not? Because they're not the same thing, right? So the metabolic rate of a bug has nothing to do with burning energy for heat. Does that make sense? But that's actually a lab that we do at the end of the year, because we can look at that, at the actually heart rate of a bug compared to the heart rate of you, right? On the same temperature, right? But you can't compare the bug, but the mouse you could, because it's doing something different. The cell respiration lab that's coming up is a play into that. You have respirometers where you can actually use that to measure the consumption of carbon dioxide, which is directly related to the metabolic rate, right? Now, if you use an invertebrate inside a respirometer, and you compare that to a mouse inside not a mouse, but a vertebrate inside a respirometer, endotherm, you would have a different number. Does that make sense? And the virtual lab is about that. You can't actually do mammals in here, but the virtual lab lets you do that. So make sure you look at that. Excess of free energy, right, versus required free energy is what we. Okay, so to have growth, think about growth. It's easy to understand. When does growth happen? Sideways growth. When there's an excess of energy. The same thing is true about population growth. So excess equals growth, right? Meanwhile, requirements mean shrinking. Does that make sense? That's simple. And then the last thing, insufficient acquired free energy versus the required free energy results in loss of mass and ultimately death. Some ordinary plants are falling apart. They're not getting enough energy or enough nutrients or something that they need to do what they need to do, right? And that's why you have to be careful. All right, so that's 2A1. The next one should be 2A2. So let's talk about 2A2. Now, the thing about 2A2 is that I need an hour for it, and we only have 20 minutes left. That's not going to happen, right? So, but 2A2 is about photosynthesis and cell respiration and all the processes associated with it, okay? So, again, make sure you watch those animations for the stages because we're not going to be able to finish it. We also, technically, have to look at... Actually, going the wrong order, I think. I think before 2A2, it's... Oh, yeah. We also technically have to do a 4B2, but I'm not going to get that far. But let's talk about 2A2. All right. Autotrophs capture free energy from the environment. That's what they do. But there are different types of autotrophs, right? You got the ones that do it based on chemical reactions that do not require sunlight. Most of them still make sugar. It's carbon-based chemistry, right? But they use the free energy from other molecules present in the environment. There will be halophytes, thermophiles, other types of bacteria that live in these extreme environments are classic examples of that. And we think that life's original strategy was chemotrophic, right? That's why we say they're most similar to the ancient life forms, although they're technically evolved from the ancient life forms. So don't think of archaeas as the ancient life forms. They're just similar to what we think the ancient life forms would be like, but they've been evolving since the start, right? Now, photosynthetic, of course, uses the sunlight. That's stupid. Okay. Heterotrophs, on the other hand, they still capture energy too. Everybody's going to capture energy. By the way, how does everybody, 99% of life, get energy? From what? So when you make energy because you need it, either to warm yourself up or to do anything, what is it really doing? What process is the primary energy driving process of the world? Which specifically? Glycolysis, 99%. Everybody does it. I want you to understand this. Cell respiration is how everybody gets energy. The difference is how you capture it. Are you with me? So phototrophs also do cell respiration to get energy. But they capture that energy through photosynthesis to make the sugar they burn themselves. We, on the other hand, as heterotrophs, must eat in order to get that energy. So we catabolize carbohydrates, lipids, proteins, nucleic acids. By the way, that's another thing. Misconception. People think that cell respiration is the destruction of sugar. That's actually not true at all. There's a picture on the book that talks about this in more detail, so make sure you look at it. But the mitochondria is an ultimate gateway for the breakdown of all organic molecules. Now, so in Introduction to Biology, we start with glucose, we do glycolysis, and we get to an intermediate molecule, right? It's called pyruvate, right? And then you go into the mitochondria with that pyruvate, and then so this pathway of carb breakdown, by the way, which means if you start from fructose or galactose, first you're going to convert it to glucose. Or convert to something here in the between. Are you with me? So let me try to find these this diagram. Metabolic pathways. I know I have a picture of it somewhere, but I'm going to try to. This is a crazy diagram of all the metabolism that happens inside of one type of cell. Look at that. It's a flow chart of metabolism in type of a cell. Yeah, I see a couple cycles in there, so I would be surprised if that's like maybe a Krebs cycle in there, you know? But that shit crazy. Anyways, that's what I want to show you.
Just a little bit, just.
It's such a shame because I really want to have the specific one that I want to look at. I think it's in my lecture series. It's worth the time because instead of drawing and talking about something, I'll have some specific. Okay. Classes. Unit 5. See, this is why I can only be as good as the time I have. I should have this prepared, but unfortunately, I teach 6 preps, so I have no time to prep it. Alright, so then, it's got to be here somewhere though. Pretty sure this lecture has it at some point. Is this cell reproduction? What am I doing here? Need energetics. Cell respiration. Presentations. Video lecture. Alright. So, by the way, this is just trying to show you that the majority of the energy that we burn goes to heat. On purpose, though. We actually want to waste that to heat ourselves up. This is another one that's super interesting, and it kind of talks about something that's important on that equation there. Notice that glucose is made by the chloroplast and then consumed by the mitochondria, right? Now the mitochondria breaks the glucose down to carbon dioxide, which means we don't make carbon dioxide out of oxygen. We don't. There's a big misconception that we put oxygen in and put carbon dioxide out. This is coming from sugar. So we eat food and that food becomes air. Does that make sense? Meanwhile, we drink water. No, we pee water. That water is coming from the oxygen we breathe in. So the oxygen we breathe in goes out as heat. And the carbon dioxide that you're putting out is coming from the food you eat. Does that make sense to you? Misconception. Likewise, plants, they're built of cellulose. That cellulose comes from air. Carbon dioxide gets trapped into cellulose. Are you with me? The water that comes from the roots leaves as oxygen in the air. Does that make sense? So cellular respiration breaks down glucose to make carbon dioxide, which is then captured by the chloroplast to repeat the cycle. Meanwhile, there's a little cycle of water and oxygen. Water is broken down by it's produced by the mitochondria, used by the chloroplast to make oxygen out of it. So it's a misconception people have, and I like to show that. Either way, we certainly play in the carbon cycle. And yes, in a way we are taking carbon dioxide out and putting oxygen in. But not one to the other. Does that make sense? In between, there's something that happens. Okay, no time. All right, seriously. So these are the processes of cellular respiration. We'll get back to them in a second. But I just want to show you that first thing about, and of course it's not in there. Okay, so then you're going to have to look at your book. There's a picture at the end of the chapter, okay, that talks about the metabolic pathways. Some lipid pathways, when you're breaking down, say, the glycerol, right, dump here. Other lipid pathways dump in between, the acetyl-CoA. Other lipid pathways dump here. But lipids can be broken down too. Proteins can be broken down too. And they all dump at some point in the pathway. Do you understand what I'm trying to say? So we break down all macromolecules for energy. We prefer to break down glucose. But when the glucose is not around, we start destroying fat to make energy. And when the fat is not around, we start destroying what? protein, muscles to make energy. That's why anorexic people look like the way they do, because you start breaking. Does that make sense? All right, I wanted to point that out. Okay, going back over here. Fermentation. So if you look at fermentation, all right, what fermentation is, in the absence of oxygen, where you cannot complete the rest of the process, because the mitochondria, by the way, mitochondria has its own internal ribosomes, internal DNA. And it's like a living cell. We talked about this already. Now, glycolysis produces pyruvate. Now remember that lipid pathway and protein pathways can enter at different points of the cycle. But glycolysis happens in the cytoplasm without oxygen. It's anaerobic, ancient. 99% of life does it. Evolved before photosynthesis, based on breakdown of sugar produced by chemotrophic pathways. So this precedes photosynthesis. This is after photosynthesis, because this is actually the reverse of photosynthesis. So what I usually talk about is glycolysis first, then I stop, teach photosynthesis, and I come back and teach this part, because that's the order in which it evolved. But what I'm going to talk about something is that happens is that glycolysis produces this pyruvate, but we can't do anything to the pyruvate, right, if we don't have oxygen, because all of this ultimately requires oxygen in the ETC. So if you don't have oxygen, you're going to there's no point in doing the Krebs cycle, because you're going to do these electron carriers, but the electron carriers are only important for the ETC. So if you make these electron carriers, but you can't do the ETC, it's like a waste of energy. So the cell stops that. Now, there is a little production of ATP, not through the ETC, which is produced by enzymes in liquid. The ETC is literally a hydroelectric dam of sorts. So it's nothing to do with chemistry so much as it is to do with physics. All right? This, on the other hand, it's basically osmosis that's happening here. Diffusion. We're going to talk about that later. Not today, I don't have enough time. But this one is chemical level substrate. Like enzymes are doing this job more. In the substrate, in the liquid. Now these things are producing ATP, so they are actually going to answer your question. Right? So now, this produces a very little ATP. That produces a lot. So this produces, per molecule of glucose, 2 net ATP, which actually get used up in this arrow right here. So you don't actually make any of it. Now this one produces 2 ATP per molecule of glucose. So after all of that, you don't have a lot of energy still, but you need a lot of electron carriers, and they dump into the ETC to produce 36 ATP. And that is where the magic happens. And after that evolved, you have the Cambrian explosion, because life can do a lot more because it has a lot more energy. Right? Until then, we were only doing glycolysis. But the thing about it is, without oxygen, you can't move forward. So what do we used to do? Instead, by the way, this is glycolysis. You do not need to know the intermediate steps and the enzymes associated with glycolysis. But notice something interesting. You actually have to invest 2 ATP to charge the glucose molecule. And then it breaks down into 2 molecules, which then get modified in a series of steps to produce ATP again. But since this is 2 molecules, this is going to happen twice. So you actually invest 2, but get 4. So you get a net of 2. And notice also that you also produce an electron carrier somewhere, right there. That electron carrier. So you don't produce only the 2 net ATP, you also produce an electron carrier. But the electron carrier is useless without oxygen, because you can't do the ETC. So fermentation is actually about that. If you look at what's happening in fermentation, you have pyruvate because you did glycolysis. So this is glycolysis, right? So I produce the 2 net ATP. Now I have pyruvate and this thing here. I can't actually use that, though, without the ETC. So what that's all about is about regeneration. This is the thing. I can keep doing glycolysis with glucose and keep making pyruvate and keep making 2 ATP, but not if I run out of these guys. Are you with me? If I run out of NAD, this guy over here, I can't keep doing this. The problem is that the ETC is supposed to use up the NAD and put it back here. But I don't have oxygen. So I run out of NAD. Does that make sense? So fermentation does not actually produce energy at all. It's all about doing what? Recharging or getting the NADH back to NAD. Does that make sense? So this is actually an endergonic process. It's actually consuming the energy that's inside of the electron carrier. All right? So that you can we build a decharged electron carrier so that glycolysis can happen again. So when you have no oxygen, all you can do is the glycolysis and get 2 ATP at a time. Are you with me? But you can't do it if you run out of the electron carrier that you need to do it. And so fermentation is just about recharging or reproducing that, recycling that. Does that make sense? Because this is supposed to be recycled by the ETC. Before the ETC is around, that's how cells got energy. Right? It was like, that's it. That's all it could do. Which produces a lot of lactate. Now the lactate does not cause cramps, but the byproduct of dealing with the lactate causes cramps. So that's when we don't have enough oxygen going through our muscles. We get cramps because we're not breathing at a rate that's fast enough to deliver oxygen fast enough for the cells to do the ETC. It could happen in your sleep too. All right. Now, is everybody clear on this? There's only a step that's actually consuming. Well, let's talk about electron charging here. If I am a carrier, that means I want to be receive electrons. Now, when you receive electrons, you become charged. Okay, that's called oxidation. All right. So because you get a negative charge, I think of it as literally it's a battery getting electrons in it. So the battery is becoming charged. Does that make sense? So I didn't have the electron. Now that I have it, I can make a bond to a hydrogen. So see, that's how it is. Once it becomes charged, I can't just capture the electron. I have to capture it in a bond. Does that make sense? That bond that I make with the hydrogen, though, is only possible if I have an electron to share with the hydrogen. Are you with me? So the hydrogen wants, I'll come with you as long as there's an electron I can share with you. That process is called oxidation. And that happens a lot in these processes. Every time a molecule receives an electron and it makes a bond with the hydrogen, it becomes charged. Because now it can give up that electron and break that bond with the hydrogen to give up that energy. Does that make sense? That would be calling decharging the battery. So if you look at it, what fermentation is actually doing is decharging a battery, right? So that you recycle the battery so you can keep doing this. Because the point is to make that. But you can't do that if you don't run out of this battery. Because if you look at the pathway of glycolysis, that battery is necessary right there. So this is not going to happen, which means all of this doesn't happen, which means you don't get the 2 ATP back. Does that make sense? So you need that. So that's glycolysis. Ancient. Parafermentation, also ancient. Because from the beginning, they were working together to create energy for the cell. But that's a miserable amount of 2 ATP. Right? And so we've got to have something better to create energy. But you're going to have to wait for that because we're going to have to talk about photosynthesis first. All right? Going back to the thing. All right? Along the same lines, okay, it says here that photosynthesis and cell aspiration rely on electron carriers. For example, in photosynthesis, there's a different electron carrier, a cousin of the NAD. It's called NADP. That electron carrier is a receiver of electrons. Now, there's also the FAD and the NAD in photosynthesis, cell aspiration. And there's also oxygen. Because at the end of cell aspiration, the ETC is a final acceptor of electrons, because this is very important. At the end of the ETC, there's an oxygen hanging out, waiting for electrons. Because that's why it's called oxidation, because oxygen is the queen. I talked about this. It wants to steal those electrons, right? It sits there at the end of the ETC saying, give me electrons. Now remember what happens. You can only make bonds to electrons that you decide to bond with hydrogen, so you make water. Because you're putting H2O. You see what I'm saying? Each oxygen wants 2 electrons. So it takes 2 electrons, 2 hydrogens, and forms water. Which means the ultimate electron acceptor in cell aspiration is oxygen. So, but there's also these intermediate electron carriers like NADH and FAD, right? So, and in photosynthesis, there's another one that's called NADP. So you have oxygen to remember as an electron carrier or acceptor. You have this electron acceptor in photosynthesis. The P reminds you of that. And in cell aspiration, you have NAD and FAD, right? FAD plus. The plus means they need an electron. They're in need of one, right? Okay. Now we have to go back to photosynthesis to talk about the process that actually creates the energy, and then go back to cell aspiration to talk about the process that uses that energy back. But we only have 2 minutes left, so I'm not even going to attempt it. Now, make sure, I'm going to show you something that I need you to do. I'm going to send you a video a link to this video because, actually no, you know what? I'm just going to show you where it is. So if you go to Unifiles and you go to cytology, I need you to do this because I'm not going to have enough time. The next time I'm going to have for our lecture is a week and a half from now, and actually 2 weeks from now, right after the spring break. So if you look at the calendar, no, the Thanksgiving, which means we're going to table this talk about energy for a little while, right? But anyways, if you look at this folder right here, cell energetics, there's two: cell aspiration and photosynthesis. I'm going to go to cell aspiration just for the sake of argument. Go there and go to videos and web tutorials. As you can see, the animations in biology are right there. Now, these are excellent little videos. They will walk you through each of the pathways: glycolysis, the ETC, Krebs cycle. Are you with me? In addition, you see the bioflicks? They're really good as well. So take a look at those bioflicks. And in the photosynthesis one, they also have similar ones. All right? And look at those videos and study with them because I'm not going to have enough time to do what I usually do, which is talk about it in a lot of detail. All right? But on the next time we have a lecture, we will talk about photosynthesis and cell aspiration. I'll see you guys then. We are actually approaching the end of the material that is actually associated with the next big idea test. Except the actual big idea test only after we come back from the winter break. But yeah, I'm just postponing the test, but for the sake of the labs. But the material we have to finish it is this plus 4B2. So yes.
I came back after school yesterday to take my test on my computer and you weren't here.
I know.
But you told me that I had to take it Monday or I wasn't going to take it.
Oh shit! I forgot about you. When do you want to do it then?
You can't stay today either because you shot it out.